Welcome to the Deep Dive. We've got quite the journey ahead of us today, folks. We're diving deep into the world of genetic engineering and CRISPR. It's pretty mind-blowing stuff.
Absolutely. We're talking about a tool that could rewrite the future, literally.
Yeah, rewriting the future. That's a good way to put it. But before we jump into the deep end with CRISPR, maybe we should take a step back.
Sure. To really grasp the impact of CRISPR, you got to understand the bigger picture of genetic engineering.
Okay, so genetic engineering 101.
Right. You know, we often think of it as this cutting-edge science, but humans have been manipulating genes for millennia.
Oh, yeah, like way before we even knew what genes were.
Exactly. Think about it. Farmers selecting the best crops or livestock to breed, passing down desired traits over generations.
So like breeding those super juicy tomatoes or those fluffy sheep.
Exactly. That's genetic engineering in its earliest form. We've been shaping life for ages.
Never thought of it like that, but it makes sense.
And all that without even knowing the science behind it.
So when did we start really cracking the code of life?
Well, one of the biggest breakthroughs was in 1953, with the discovery of DNA structure. Watson and Crick, and of course we can't forget Rosalind Franklin's crucial role in that.
Right, Rosalind Franklin. She doesn't always get the credit she deserves.
True, but her work was instrumental.
Okay, so 1953, the blueprint of life is revealed. What happened next?
Then, boom, 1973, Boyer and Cohen successfully transfer a gene from one bacterium to another, the first time we directly manipulated genetic material.
Okay, things are starting to get really interesting now, so that leads us to...
Genetically modified organisms, GMOs.
Right, GMOs. They're kind of everywhere now.
And that was just the beginning. Fast forward a few decades, and we enter the era of CRISPR.
CRISPR, okay, so this is the big one, the game changer, right?
Oh, yeah. This is where things get really wild. Imagine a bacterium constantly under attack by viruses.
Sounds rough.
It is. So bacteria develop this incredible defense mechanism, CRISPR.
So it's like a bacterial shield.
More like a memory bank. When a virus attacks, the bacteria stores a piece of the virus's DNA in its own genome.
It remembers its enemies. That's pretty clever.
Right. So the next time that same virus tries to attack, the bacteria is like, nope, not today.
So it uses that stored DNA to recognize the virus.
Exactly. And then it unleashes this enzyme called Cas9.
Cas9, is that like?
Think of Cas9 as molecular scissors. It uses that stored viral DNA as a guide to cut up the invading virus's DNA.
Whoa, it slices and dices the enemy's DNA.
You got it. It's super precise, super effective.
Okay, so we've got this bacterial defense system, but how did we go from that to gene editing?
That's where brilliant minds like Emmanuel Charpentier and Jennifer Doudna come in. They realized, hey, what if we could control what those molecular scissors target?
So instead of targeting viruses, we could target anything.
Exactly. They figured out how to create a synthetic guide RNA, gRNA for short, that matches any DNA sequence you want to target.
So you're basically giving Cas9 a new target.
Yep, you're telling it, go cut this specific gene.
Okay, so now we can target and cut specific genes.
And the cell's own repair mechanisms kick in, either disabling the gene or allowing you to insert a new piece of DNA.
So you can disable or rewrite genes. That's huge.
Huge is an understatement. We've been able to edit genes before, but CRISPR is different.
Okay, so what makes CRISPR so special?
It's faster, cheaper, and way more precise than previous gene editing techniques. It's like trading a clunky old typewriter for a supercomputer.
So CRISPR is the supercomputer of gene editing. That is wild.
It's completely revolutionizing the field, opening doors we never even thought possible.
So now we can edit genes with incredible precision, and that brings us to...
a whole new world of possibilities and some pretty big questions.
Yeah, that's what we're going to dive into next. So a whole new world of possibilities. Where do we even begin?
Well, let's start with medicine. The potential is just mind-blowing. We're talking about curing genetic diseases. Think sickle cell disease, beta thalassemia.
Diseases that have plagued people for generations.
Right, and they're caused by just a single gene mutation. With CRISPR, we can potentially correct that faulty gene, like, permanently.
That's incredible, like a cure, not just treatment.
Exactly. And we're already seeing promising results in clinical trials. They're editing genes in blood cells, and some patients are seeing their symptoms completely disappear.
Wait, so people with sickle cell disease are living symptom-free?
Some of them, yeah. They're no longer experiencing those excruciating pain crises or the complications that come with the disease.
That's life-changing stuff. It gives me goosebumps just thinking about it.
It really is remarkable, and it's not just limited to sickle cell disease.
Oh, right. There's so much more. What else is CRISPR doing in medicine?
Well, it's also making huge strides in cancer treatment. There's this thing called CAR cell therapy, where they basically engineer a patient's own immune cells to fight cancer.
I've heard about that, but how does CRISPR fit in?
CRISPR makes those engineered immune cells even more powerful, more precise. They're like supercharged cancer fighters.
It's like giving your own body the tools to fight back.
That's a good way to put it. And there are even trials using CRISPR to correct genetic mutations that cause blindness.
So potentially restoring sight to people who have lost it.
Exactly. It's like something out of science fiction, but it's happening right now.
Okay, wow. CRISPR is like this miracle tool, curing diseases, restoring sight, fighting cancer. It's incredible. But with that kind of power, there have got to be some ethical concerns, right?
Oh, absolutely. The power of CRISPR is a double-edged sword.
So what are some of the big ethical challenges we're facing with this technology?
Well, one big one is the idea of designer babies.
Yeah, I've heard about that. If we can edit genes for disease prevention, what's to stop us from editing for other traits, like intelligence, athleticism?
Or even physical appearance. It's a slippery slope.
Where do we draw the line?
Exactly. That's the question we have to wrestle with as a society.
Because if we start selecting for desirable traits, that could lead to some serious inequality.
Right. Imagine a world where only the wealthy can afford to enhance their children's genes. That could create a whole new level of division and discrimination.
It's a scary thought, not just about humans, right? What about the ecological impact of CRISPR?
That's another big concern. Genetically modified organisms getting out into the environment, potentially disrupting ecosystems, creating invasive species.
It's a lot to consider. We have to be really careful with this technology.
We do. We need clear guidelines, regulations, open discussions about the ethical implications. We can't just blindly rush forward.
We need to find that balance between innovation and responsibility.
Exactly. It's not an easy task, but it's crucial.
So where do we go from here? What's next for CRISPR?
Well, the research is constantly evolving. There were all these new, even more precise techniques emerging, like base editing and prime editing.
CRISPR 2.0, huh?
You could say that.
Yeah.
Base editing lets us change a single letter in the DNA code with incredible accuracy. And prime editing takes it even further, allowing for more complex and controlled edits.
So we're talking about even more precise, even more powerful gene editing.
We are. And that opens up a whole new realm of possibilities, for better or for worse.
It's both exciting and a little bit terrifying. What are you most excited about in terms of the future of CRISPR?
Well, one area that really fascinates me is the potential to reverse aging.
Reversed aging. Is that even possible?
It might sound like science fiction, but it's an active area of research. Scientists are looking at using CRISPR to target genes that are involved in cellular aging.
Wow, that would be revolutionary. And what about space exploration? I've heard rumors about CRISPR being used to, help astronauts adapt to space.
It's not just rumors. Think about the challenges of long-duration spaceflight radiation, bone loss, muscle atrophy. CRISPR could potentially help us overcome those challenges.
So we could engineer astronauts to be more resilient to the harsh environment of space.
Exactly. It could be a game changer for space exploration.
It's incredible to think about all the possibilities CRISPR unlocks. But it also feels like we're stepping into the unknown, into uncharted territory.
We are, and that's both exhilarating and a bit daunting. It's a powerful tool, and we need to use it wisely.
That's a lot to take in, you know? Curing diseases, enhancing crops, space travel, reversing aging. It's like we're on the verge of reshaping life as we know it.
Yeah, and CRISPR's at the heart of it all. It's making us question, what does it even mean to be human?
If we have the power to rewrite our own DNA, are we playing God? It's a question that goes way beyond science.
It delves into philosophy, ethics, morality. It's a conversation we all need to be a part of, not just scientists.
We need everyone at the table thinking about the implications, weighing the benefits and the risks. Because this technology, it could change everything.
And we need to make sure those changes are for the better, for everyone.
One thing that worries me is the potential for CRISPR to make inequality even worse.
Oh, yeah, that's a huge concern. Imagine a world where the rich can afford to genetically enhance themselves and their kids while everyone else is left behind. It's a recipe for disaster.
A genetic divide. That's a scary thought.
We have to be really mindful of those societal implications. CRISPR needs to benefit all of humanity, not just a privileged few.
And then there's the whole issue of unintended consequences. Messing with genes, It's not like rearranging furniture, it's complex stuff.
Absolutely. We're still deciphering the intricacies of the human genome. You tweak one gene, who knows what effect that might have on other genes or the whole organism.
It's a bit like playing with fire, isn't it?
Yeah.
We need to be incredibly careful, incredibly responsible.
We need to proceed with caution and humility. We don't have all the answers. More research is needed, and open and honest discussion is crucial.
It feels like we're explorers venturing into uncharted territory. There's so much potential, so much to discover, but also so much that could go wrong.
And that's what makes it both exciting and daunting. We're at a crossroads, and the choices we make today will determine the future of humanity. CRISPR is a tool of immense power, and like any powerful tool, it can be used for good or for ill.
Well said. I think that's a good place to wrap things up. This has been quite a deep dive. I'm walking away with a sense of awe and a whole lot to think about.
I feel the same way. This technology has the potential to reshape our world in profound ways. It's up to us to make sure we shape it wisely.
So to everyone listening, thanks for joining us on this journey. Keep asking questions, keep learning, and keep the conversation going. This is a future we're all building together. And until next time, keep exploring.